Now you just go through the diagram showing homologous chromosome. The two homologous chromosomes, one is blue in color, another one is red in color. So they are having same size and same shape. In the first diagram it is given there. In the second one, I think you have noticed that the part of chromatids, blue chromosome consists of two chromatids. These chromatids, you know, are called sister chromatids. And what is the relation between one chromatid of blue chromosome and one chromatid of red chromosome? Their relation is non-sister, non-sister chromatids. In the second diagram, non-sister chromatids, they exchange their part. And in third diagram, recombination of chromatids. See, the blue chromosome, one chromatid of blue chromosome having some portion which is red in color. In the same way, red color chromosome having blue color. It occurs due to the exchange of part of chromatid. And two terms are related with this process, that is crossing over and chiasma. So once again I am starting from homologous chromosome. Homologous chromosomes are a set of chromosomes, one from mother and one from father, that pair up with each other inside a cell during meiosis. They have the same genes at the same loci, same genes at the same loci. Now crossing over. Crossing over, the non-sister chromatids, there is blue and one red chromatid. So the non-sister chromatids of tetrads seem to cross each other at one or more points. Here only one point is showing, but it can be occurred in more than one point also. These points look like X-shaped structure. This X-shaped, I think you have noticed that the place where red and blue chromosome exchange red and blue chromatid exchange their part, the portion is looking like X-shaped structure. This structure is called, this point of crossing is called chiasma or chiasmata, singular and plural. And the process of exchanging chromosomal part is called crossing over. These two definitions are given. First one is crossing over, the exchange of chromatid segments between two non-sister chromatids of homologous pair is known as crossing over. And chiasma, the point of interchange of non-sister chromatids form a X-shaped structure which is called chiasma. Now I am going to discuss about cell cycle. Cell cycle, the definition is a sequence of events that occur in a cell in preparation for cell division is known as cell cycle. It has four phases: G1 or gap 1 phase, S or synthesis phase, G2 phase or gap 2 phase, and mitotic phase. You just go through the diagram. Blue portion, total blue portion including G1, S, G2. These are the divisions of interphase. That means interphase has three parts mainly: G1, G1, S, G2. All are including in interphase, then mitosis. That means after completing mitotic cell division, I have discussed already, after completing mitotic cell division by prophase, by maintaining prophase, metaphase, anaphase and telophase, by completing prophase, metaphase, anaphase and telophase, two daughter cells are produced. These two daughter cells take rest or enters into interphase for growing in size, for the replication of DNA, for the synthesis of protein, etc. It takes time. This resting time or resting phase is called interphase. That means interphase has three subphases, that is G1, S, G2, and then again the cell will start mitotic cell division. So I am giving by points. What happens during G1 phase? G1 is the first gap phase or growth phase. It is the preparatory stage for oncoming divisions. I mean, to take part in the next cell division. The cell grows in size during G1 phase. Cell grows in size, and besides synthesizing RNA and proteins, the cell grows in size besides synthesizing RNA and proteins. No change occurs in its DNA content. There is no change in DNA content during G1 phase. G1 phase is most available. G1 phase is most available with regard to duration. I mean, this is the longest phase. Then synthesis phase. During this phase, replication of DNA and synthesis of histone protein occurs. The chromosomes are duplicated. Then G2 phase. This is the second gap phase or second growth phase of interphase. In this phase, synthesis of RNA and protein continues, which is required for cell division. Two centrosomes are formed by replication of a single chromosome. As the G2 phase draws to a close, the cell enters into a mitotic phases. That means after completing G2 phase, the cell enters into the mitotic phases, that is prophase, then continuously metaphase, anaphase, and telophase. Now I have completed cell cycle also. Here assignment is given. You have to complete all the experiments given in your lab manual with diagram. It has to be completed in your practical notebook, biology practical notebook.